and we go to the last presentation, and that is from Professor Pim Pijnappel from Erasmus University, and he leads the translational lab at our center, and he's going to talk about stem cell and muscle regeneration in Pompe disease.
All right, thank you. This is working? Yes. I will start with some work on stem cells. So in the muscle we have actually adult stem cells, and these are called satellite cells. You see here a cartoon of a muscle fiber, and satellite cells are actually close to the fiber. They are in between the basal lamina and the plasma membrane of the muscle fiber. And if you injure your muscle, or you have diseased muscle, the satellite cells can repair muscle, and they can fuse with the existing muscle fiber, and then they can rebuild your muscle. And this is a very powerful system. So in skeletal muscle we have a very powerful regenerative capacity to repair muscle. And that of course is very interesting for muscle disease. The question is, what happens in muscle disease, and can we exploit that to build new muscle when the muscle is lost due to the disease? That's something that interests us a lot, and it is extremely difficult, I have to say. But the biology tells us that at some point this should be possible. There are a couple of challenges that we need to overcome to make this really a viable therapeutic approach. Here you can see actually what I was just mentioning, and I hope the video is going to play. Yes. So here you can see actually muscle cells that are fusing together to make muscle fibers, and that is just to illustrate what happens if your muscle develops. And before going on, I will also introduce you to the human iPSC technology. And this is also a lot of technology that I will present to you today that we can hopefully use to make new steps in therapy development. And human iPS cells mean induced pluripotent stem cells, and that stands for iPS. If you look at the left, these represent normal cells in an adult individual. For example, skin cells from a skin biopsy, or blood, or urine, or teeth, or any cell from your body. And there was a Japanese professor, Yamanaka, who did a crazy experiment. Actually, in his lab, a crazy experiment was done. And actually he went back in time. He went back in time to make from adult cells embryonic cells, embryonic stem cells. And you would say, when you would start this experiment, don't do it because it will never work. But actually it worked in a very simple way, and now the whole world is using it. And it's called dedifferentiation. So you go back in time, you make embryonic stem cells, like cells. But they are not really embryonic stem cells. They are similar to embryonic stem cells. They are created in a lab, and therefore they are induced. They are pluripotent. That means they can make all cells of the human body, as you can see on the right. So they can make neurons, or muscle, or bone, or cartilage. And that is a new technology that is now very useful, because now we can actually make human cells, tissues in the laboratory. We can make them from patients with a very simple procedure. And by this technology we can move from the mouse to the human, and we can replace animal experiments, but we can also look actually in patient-specific way how therapies would work. Of course, then there's a huge challenge. How can we actually rebuild the tissue that is relevant for the disease? In this case, muscle. How can we rebuild muscle? And that's what part of this talk is about. And that's what we are trying in the lab. And there are several stages that we went through. So you start with making cells. So making iPS cells, that's a Nobel Prize winning technology from Professor Yamanaka. But then to make muscle cells out of the embryonic-like cells, or the iPS cells, it turned out to be quite difficult. So this was the first hurdle, but we took it. And then in phase 2, we actually developed some technology to make the 3D muscle tissues from those cells. And we optimized this technology in several ways. I will show you that. Also to make disease models, so you can then model a disease like Pompe disease or another muscle disease and try to recapitulate the disease in the laboratory. If you want to make this really applicable to the testing of therapies, of novel therapies, you want to increase throughput. So we, in parallel, developed technology together with a company, Optics 11 Live, to measure multiple muscle tissues in real time. So now you can really do a proper experiment. And then we moved to some drug testing to further see how the human muscles would respond. So to start with the cells, there's a procedure that we are now using in the lab that we published, so you can all read it back in 2018 already. And somehow we found that we could also expand the cells to huge amounts. This is really an advantage, because if you take cells from a biopsy, you cannot really expand them. Somehow they gave up, but these cells that we created with specific medium conditions, we can expand to large amounts. So then we can purify them, we can make them from iPS cells, and then we can do many, many experiments. From one purification, we can actually make one PhD student very happy for 4 years. So just to indicate that. And you can see some pictures. These are cells grown on a plate. So some muscle fibers that we can make from these cells if you further differentiate them, and some stainings, and you can also see the contractile unit, the sarcomere. You also see it's pretty disorganized. They all grow across each other. And for certain experiments, this is very useful. But for other experiments, you really want to recreate the native environment of a human muscle tissue. But we already started also to do some regenerative experiments. So we used these cells and injected them in a mouse that was pre-injured. So you actually create a momentum by doing that. So you pre-injure a muscle, and then you inject these cells that I just described to you. And these are human cells in mouse. And we were indeed able to make human muscle tissue in the mouse. So this is a proof of concept that these cells that we had are actually able to form human muscle tissue. And that, of course, is the first step in a regenerative project where you could actually recreate human tissue that has been lost due to disease. And what you see here is several stainings, and it is an advantage. If you use human cells, you can use human-specific antibodies that recognize only the human protein. So that's what we did. In left in color, and on the right we made it black-white to make it a little bit more clear. And you can see everything that's white there is human. So you can see either nuclear staining, these are the dots, or stainings of the plasma membrane, and these are actually the circles. This works, but it's not efficient enough. To make this really important or clinically relevant, we really need to increase the efficiency. And we are working on that. We are not there yet, but at some point I hope we will make it at a higher efficiency. So we continued also to make muscles in the laboratory for in vitro testing. And you will not find this publication in PubMed, but this is a more technological journal that we published this work in. And actually here we used a 3D printer to make little chambers where we can grow 3D muscle. So on the right bottom, you can see a few of pillars that we use. These are the tendons actually that we recreated using a 3D printer. And between those tendons, we grow 3D muscle tissue. And on the left you see a complicated picture of, you can forget it, but this is the way we 3D print a mold, and we make a replica of that, and the replica has the material in which we can grow the muscles. And you see the pillars that we can make, and these are 3 sizes. So we can make them very small or medium size or a little bit larger. And yes, we have actually this is the way to cast the muscles. So we have a hydrogel, we add cells, and this is an artificial extracellular matrix that we are using to and we're putting the cells in, and they kind of self-organize. And you can see here an example of such a 3D muscle. In this case, we did a live calcium imaging. Calcium is a very important signal that that is used for contraction. So you get an influx of calcium in the cells if the muscle is contracting, and you can visualize that here. So if it lights up green, that's actually the calcium signal. And on the right, you can see the muscle fibers that we recreate in 3D. They are nicely parallel, so they self-organize between these two tendons. And you can also see that we also can create muscle stem cells that we can stain with a marker for PAX7. So this is a very nice model system to make human muscle, and this is actually we can make from each individual in this room via, for example, a skin biopsy. And then you get like personal testing of therapies per individual. This is the way to, in this system, because we have another system, this is the way to measure the force. So you use a microscope and you can look at the pillars. So if you put electrodes that you can see there with an Arduino circuitry, you can put an electrical pulse in a medium, and the pillars are flexible. So the muscle starts to contract and starts to move the pillars to each other. And you can measure the movement. You can make a video, and then you can calculate the force based on the pillars. And that's actually the formula that you then use for that. And it's a pretty, you just fill in the formula and you have your contractile force. We went on with applying this technology using 3D printers, and we published also recently in this 2023 paper the forces that you can generate with this kind of mini muscles in 3D. And you can see three individuals, control 1, control 2, control 3. There are some differences always. Of course, there are differences between individuals. Some people are strong, some people are less strong. And we also made a comparison to primary cells from a muscle biopsy. So if you take a muscle biopsy, you have a short window where you can do a proper experiment, and then you lose the cells. And that's what we did, and the forces that we use there are quite comparable. And on the right you can see a proteomic analysis also showing that they are quite comparable. We also looked at disease modeling with a different method. So we are using CRISPR-Cas9 also, but now to model diseases. So we can introduce a mutation, you can correct a mutation, you can make pairs where you have a healthy and a diseased version of the same individual using CRISPR-Cas. So that's very important to control for genetic background. Each person is different, so you have to correct for that. But it takes a lot of time. It takes like 2 months to make the myogenic cells that we are using from a patient, and then it takes also 2 months to do the gene editing, and it takes 2 months to further make the cells. And in this way, this is a knockdown strategy. So here we knock down the gene instead of doing a knockout. So we don't make a mutation, but we just reduce the amount of messenger RNA using shRNA. And usually if you do that in 3D tissues, it's extremely inefficient. But we mixed in the technology in the hydrogel. So with a specific method, we were able to get a very high efficiency. And we were able, and you see that actually on the low side with the green dots, that's just showing that we can enter the complete 3D muscle with this technology. And we could actually model, in this case, 2 diseases, Duchenne muscular dystrophy and Kelpen 3 for limb-girdle muscular dystrophy 2A. And within a week, you can already see on the right that the force is completely gone. In these two cases, we also did myostatin, by the way. Myostatin were supposed to be stronger if you do a knockdown of myostatin. And yeah, I think we have to wait a little bit longer to see that effect. And on the left, you can see also the morphology, which is horrible if you do the Duchenne knockdown and also the calpain-3 knockdown. So this works really fast. And you could argue this is all non-specific, but we were able to rescue the Duchenne phenotype using the microdystrophin construct that is used in clinical trials also, indicating that's really specific and it's not like a general toxic effect. So this is another way to have a very fast disease model to test therapies. This is all quite a lot of work to do these experiments. So in parallel, we developed a high throughput method. And instead of pillars that go up, we actually used pillars that go down in the system, and we used a smart lid. And this was done together with Optics 11, the company who actually now commercializes this device. And we published this last year in the same journal that's still not in PubMed, so you can email me if you cannot find it. It's a really technological paper, but actually the principle is the same as before, but the wave detection is completely different. And here we use optical fibers and no microscope to detect the contractile force. And actually these are small light beams that can look at the pillar that is moving, and it's like 1,000 times more sensitive. And you can also actually do this for many muscle bundles at the same time. So now you don't need to take out your experiment from an incubator and make a movie one by one, and then after that look what the force was. But in this case, you can actually continuously monitor the force, make a stimulation. You can do in vitro exercise experiments, what we are actually doing right now, and you can gather a lot of information. And you can do it for multiple tissues at the same time. So that is a huge advantage. And you can actually see an example here. So you see here a contracting muscle, and on the right, parallel measurements of individual muscles. And on the left, you can see, it's called a cuore. Cuore is Italian for heart, because you can also use it for 3D heart tissue. It contains a smart lid, and all the electronics are in the smart lid, and it actually fits to a 24-well plate, a conventional plate, and there you can grow your muscles. And electrodes are also coming from the smart lid, and in this way you can easily do this in parallel. And this is another example of calcium imaging, which parallels. You can also put the whole thing under the microscope and look at live calcium imaging, so that also works. And in the last bit, I show you some drug testing. So on top, you can see the forces of an individual muscle, and we applied different drugs. First, we started with caffeine at a low concentration. These are the blue things that you see. So each bar is one measurement of contractile force, and you can see that the force increases to some extent. Then we go to a higher concentration, and you get a higher force. It opens calcium channels. Then we use a calcium channel blocker, verapamil, at a low concentration in red, and you can see the force goes a little bit down. We use a higher concentration. This is all the same bundle, and the force goes down. So this is completely impossible with the 3D printer technology. With this smart lid technology, with the parallel measurement, now you can really evaluate also the effects of drugs at a very short time frame. And this is another example. We tested clenbuterol. Clenbuterol has been used before in a mouse model. You see publication by Head in 2011 already. And in the mouse, they found actually, the peak shape was different, and there were some really strange effects of clenbuterol, but you can wash them out, and then you get the restoration of the forces. And we tested that in 3 individuals, so 3D muscles from 3 individuals. And also we found very similar results, but now for human muscles. Just another example that you can really evaluate the effects of drugs here, and you can also test, how are different individuals responding to different drugs. And I think this is going to be the future. To end up, we show here the muscle team on top, the muscle on chip team, especially Alessandro on the left. And we have also for the transplantation, we have some data I didn't show you yet. I hope we can make more progress with that when we transplanted some mouse cells, that we can actually get to a better efficiency with Gerben, Schaaf and Rodrigo. And that's actually what I wanted to tell you. Thank you.